Hello and welcome, everyone. My name is Dane Menke. I am the Digital Marketing Manager here at Regenesis and Land Science. Before we get started, I have just a few administrative items to cover. Since we're trying to keep this under an hour, today's presentation will be conducted with the audience audio settings on mute. This will minimize unwanted background noise from the large number of participants joining us today. If the webinar or audio quality degrades, please disconnect and repeat the original login steps to rejoin the webcast. If you have a question, we encourage you to ask it using the question feature located on the webinar panel. We'll collect your questions and do our best to answer them at the end of the presentation. If we don't address your question within the time permitting, we'll make an effort to follow up with you after the webinar. We are recording this webinar, and a link to the recording will be emailed to you once it is available. In order to continue to sponsor events that are of value and worthy of your time, we will be sending out a brief survey following the webinar to get your feedback. Today's presentation will focus on in-situ chemical reduction core concepts and their engineering implications. With that, I'd like to introduce our presenters for today. We are pleased to have with us Dr. Paul Tratnyek, Professor in the Division of Environmental and Biomolecular Systems and Institute of Environmental Health at the Oregon Health and Science University. Dr. Tratnyk's research concerns the physico-chemical processes that control the fate and effects of environmental substances, including minerals, metals, organic contaminants, and nanoparticles for remediation as contaminants and in biomedical applications. Dr. Tratnyek is best known for his work on the degradation of groundwater contaminants with zero-valent metals. We are also pleased to have with us today Dr. John Freim. Dr. Freim is well known throughout the environmental remediation industry for his work in developing breakthrough in-situ chemical reduction technologies. He has over 30 years of experience in materials processing and 15 years in the environmental remediation industry, and currently leads the first Regenesis state-of-the-art colloidal product manufacturing facility. In his role with Regenesis, Dr. Freim is responsible for the manufacture of colloidal materials, including plume stop liquid-activated carbon, the colloidal zero-valent iron technologies Aqua ZVI and Micro ZVI, as well as other new products used in environmental remediation. All right, that concludes our introduction. So now I'll hand things over to Paul Tratnyek to get us started.
Thank you, and good morning to everybody. This is Paul Tratnyek, and I'm speaking from Portland, Oregon. This is the first time that I've been able to use this cover slide from my office. So this is, in fact, the view from just around the corner from where we are. I'm going to give a talk today that's primarily based on the work that we wrote up in a chapter from a CERTIP book back in 2014. And I want to use this first slide here to basically make a little plug for the book and to give you a perspective on how we got started on this particular topic. So the series of books that this came from was a CERTIP sponsored series. And this volume here is the first one amongst, I think, 7 or so other volumes that CERTIP hosted. This particular volume was on chlorinated solvent source zone remediation, but the chapter that I was asked to write was on in-situ chemical reduction. And so we got together with Rick Johnson, Greg Lowry, and Dick Brown and wrote a chapter that was an effort to try to give an overall perspective and a comprehensive perspective on what in-situ chemical reduction was. For the purposes of the book, it was in the context of source zone remediation, but my interest was really broader than that. It was any application of in-situ chemical reduction. So the first part of that chapter, the technical background portion of that chapter, was framed in a general way, and that's the material that I'm going to use for the first few slides in this presentation. You can click on these QR codes if you're prepared. If not, they will come back again at the end of my presentation. All right, so the first part of this is the question of where does this terminology come from, who gets credit, and exactly how much. The term has been used for a little while, since about the 1990s, and it came up more or less simultaneously in a couple different sources. One significant one was some work by Jan Dolfing, Alan Seach, John Vogan, and others that was presented at the 5th Chlorcon meeting and is written up in an article in this Soil Sediment Contamination Journal. And their perspective on ISCR at the time was really to provide a context for marketing a few specific products that they had, which are mentioned here. They're not currently using those product names anymore as such, and so that particular version of ISCO has been essentially absorbed into the broader definition that initially was really articulated by Dick Brown, and he was talking about this in the 5th Chlorcon meeting, the 6th Chlorcon meeting, a book chapter in volume 5 CERTIP book, and then subsequently in the chapter that I co-authored with him, which we'll be using as we go. I got involved a little bit later. My first presentation on this was in the 7th Chlorcon meeting, and then this book chapter. And so my initial mark on this was to, among other things, try to give a more precise and inclusive definition to what ISCR is or should be. My perspective is non-proprietary in the sense that I don't vend any products in this realm. And I was very attuned to trying to give a consensus perspective on this problem. So I took input from a lot of people in articulating that book chapter. It's interesting to look and see how things have developed since then. Preparing this talk, I took these snapshots of what's currently on Wikipedia for in-situ chemical reduction and what's currently on the CERTIP Enviro Wiki for in-situ chemical reduction. The latter was actually written by my ex-student Min Fan, now at Geosyntec, and it still reflects our perspective as it's in the book chapter. But the Wikipedia page, I think we probably started this page, but it's since been significantly rewritten by other people. So it's kind of interesting to read and see how the wording has changed due to the influence of other players in the area. So now for a definition, I remember I was concerned with trying to give a broad inclusive definition, and so this is what we came up with. The first bullet here says, in general, in-situ ISCR refers to a category of in-situ groundwater remediation technologies where treatment occurs primarily, in italics, by chemical, in italics, reduction of contaminants. And there is a strong sense of just trying to build on the analogy to in-situ chemical oxidation here. The notion that ISCR the emphasis is on abiotic processes is fundamental, but contaminant reduction by biogenic reducing minerals is included if the role of microbial activity in the contaminant reduction is indirect. And so I should have italicized the word indirect there, because that's really the key element that defines the boundary between biodegradation, direct microbiological degradation of contaminants, and what we are concerned with in ISCR, which is abiotic degradation by reducing materials, say mineral phases. But the second bullet here is to acknowledge the fact that most of these reducing mineral phases are generated by in-situ microbiology, so biogeochemistry. And so when we have contaminant degradation by ISCR, represented by this line here, in almost many cases, the source of this is microbiology over here. The only thing we're excluding is direct biodegradation of the contaminants. That would be a different topic. We also tried to include in the scope of ISCR the idea that there's both natural and so-called intrinsic biogeochemical processes. There's also scenarios by which you might stimulate in-situ microbial activity and wind up with ISCR favorable conditions, or there's scenarios where you might engineer ISCR conditions by adding strong chemical reductants. And we deliberately designed this to try to include all of that in our scope. So when we were working on the chapter, we made a laundry list of technologies that we thought we would try to fit into the ISCR rubric, and we came up with this list. And so there's a bunch of acronyms here, some of which you all obviously will all know, PRBs, BIRD, M&A. There's a few that you'll get quickly if you are familiar with them, like this trademark version of ISCR, in-situ redox manipulation. A couple of them were neologisms of my own. This CRD stands for catalytic reductive dechlorination. I needed a name for palladium catalyzed hydrogen in situ. I also invented SCTI for source zone targeted injection, basically to give a word to how NZVI was primarily used in that context. So to put all these into a framework for the purposes of the chapter, we came up with this matrix here that's got reductant strength on the x-axis and treatment volume on the y-axis, with source zone treatment at the top here, plume treatment down here, natural reductants, so the intrinsic ones here, and engineered reductants over here. And the idea is that we can take everything here on the left and we can populate it onto this matrix, and we end up with something that looks like this. And I was a little hesitant to do this initially because you could quibble about exactly where things belong. But for the most part, people have been pretty happy with this. Obviously, abiotic MNA belongs down here under plume treatment with natural reductants. Obviously, source zone targeted injection with NZVI belongs up here under source zone treatment with engineered reductants. And these other guys here could be placed in a variety of positions approximately where we currently have them. This figure has been fairly successful. I've seen a number of people reproduce it and modify it to meet their own particular needs, and that was part of our intent from the beginning. So it's part of the roadmap that I used in developing this talk, where I extended that list a little bit. So this red column of names down here on the left is based on what was on the previous slide with a few additions. So I've added, for example, a placeholder for some source of additional type of iron 2. I'll elaborate on that in a second. I added a placeholder for polysulfide injection, iron sulfides, bimetallic sulfonated ZVI, ZVI plus pyrogenic carbonaceous material. This would be a kind of an umbrella term for activated carbon. So this list is a little bit longer than the one in the previous slide, and it presumably will continue to evolve and get longer. So it'll be a work in progress. But this part here is not really what I'm going to talk about. John will talk a little bit about that in the last part of this webinar. Obviously, the hat that I wear in this context is as an academic, and so what I'm supposed to do is help articulate, define the core concepts. And this is sort of the textbook material, if you will. And so this is my current outline of core concepts that are most relevant here. And this is also going to be an evolving list, but it was optimized for today's purpose, and you'll see we'll follow most of it more or less. When I originally started drawing this, my thought was that I was going to draw connecting lines between each technology and the most relevant core concepts, but then I got bogged down deciding which lines to draw. So I threw up my hands and drew everything connected, because the more I thought about it, in almost all cases, the core concepts apply to all the technologies. There's just a few exceptions where, for example, say in the case of abiotic monitored natural attenuation, we don't really need a line connecting that to mobility and deliverability, because you're not injecting any kind of engineered reductant. But those are the minority. In most cases, there's a connection to be drawn, and so we have this kind of disorienting looking pattern here, which is going to be, which we will cherry pick a few lines as we go. So I'm going to going back, I'm going to start here at the top. I'm going to start with mechanism of removal, reduction versus sequestration. So the ISCR can remove contaminants from groundwater by two basically distinct approaches. One of them is where the contaminants are removed by reduction. And so this would apply to organic chemicals where there are functional groups that are amenable to reduction. So the chlorinated functional groups are certainly the most familiar. Nitro groups in explosive type compounds would be another example, and that there are others. But so the main category here is chlorinated solvents. There are also metal oxyanions that are important groundwater contaminants that are subject to reduction. And so I listed chromate is probably the best known to you here, but also protectanate, selenate, arsenate, etc. There's also some non-metal inorganics that are subject to reduction that are sort of relevant here. Nitrate is certainly one of them, and perchlorate is kind of not so much, but it's an interesting exception to sort of prove the rule. In contrast to reduction, there's also the scenario that we're calling sequestration. So sequestration is an umbrella term for any kind of physical removal by sorption or co-precipitation, et cetera. And so there are metal oxyanions that may not undergo reduction, but still are effectively sequestered by ISCR type agents. And uranium would be a pretty good example of that. There's also all the heavy metal cations like cadmium, copper, mercury, nickel, palladium, or sorry, lead, which may be removed from groundwater by a process that's sometimes called cementation. And in that case, that process is mostly best thought of as sequestration. Although in all cases, many cases, there is some overlap between reduction and sequestration. And so that overlap is illustrated in a lot of cartoons that are scattered around in the literature for kind of diversity. I picked this one to decorate this slide here. It's from a review paper by Irene Lowe and Tang in Water Research. And you see here she's got iron 0, she's got magnetite, she's got iron 3 oxides, and she's got adsorption and reduction and co-precipitation. And so the point just being here that you can have both reduction and you can have sequestration contributing to the contaminant removal. So now a few slides on what the reductants are that we're talking about here. And so the first distinction I want to make are between two branches. The first branch is the relatively mild intrinsic reductants that occur naturally. They're mostly based on iron 2 and is sometimes referred to as DVI for divalent iron, and or they're composed of reduced sulfur here and here. And there's also, I got in here as a sort of a placeholder for natural organic matter, although it's not the most important in the context of what we're talking about today. There's, in contrast, or in addition to that, we have the relatively strong engineered reductants, and dithionite is certainly an example that you should all be familiar with. Zero-valent iron is the one that everybody's familiar with, and there are other zero-valent metals like zinc that occasionally can be of interest. I would point out that most of what applies to 1 here also applies to 2, so there's some overlap and some blurring between the lines. I had a bunch of space on this slide to fill up, and so I wanted a graphic. And so I was thinking, well, this notion of branches, I really got this from Dick Brown and some of his early talks around 2010 on ISCR. And so I went looking for the image that I remember from his PowerPoints that was, I think, a tree, which is presumably what the branches referred to. But this is the only thing I found. I'm not really sure what this is supposed to be, but it clearly illustrates, or it does still illustrate, the idea that ZVI and DVI are major branches of ISCR. So DVI is up here and ZVI is down here. Okay, so more on the reductants. Let's elaborate a little bit on the intrinsic reductants. So these are naturally occurring. They're mostly mineral phases that are derived from iron 2. So again, DVI. So the particular phases that are candidates for this would include magnetite, green rust, ferruginous clays, and iron oxides with adsorbed iron 2. There's also minerals that contain reduced sulfur, and so there are a number of them. McKinnoite is likely the most important. Pyrite's often talked about. Greigite, marcasite, and amorphous iron sulfides are others mineral phases that might be important in some context. And again, I have a placeholder down here for natural organic matter, but I won't develop that much in this talk. I have a redox ladder over here just to illustrate how strong these reductants are in a thermodynamic sense. So this is EH on the left and PE on the Y. Negative at the bottom is more strongly reducing. Positive at the top is less strongly reducing. So you see here the aqueous iron 2, iron 3 couple is not really strongly reducing at all. ZVI is down here, but for the intrinsic natural biogeochemical minerals, it's these guys here. And so those are the ones that are moderately strong reductants and in theory could be responsible for some contaminant degradation. reduction. There's an important aspect of this that I wanted to elaborate on that has to do with the relationship between these intrinsic reducing phases and the in situ biogeochemistry. And so this is illustrated by this very nice graphic that was done by the folks that were involved in this AFC ESTCP workshop report back in 2008. And so this diagram has got 4 parts. In each case, it uses blue for microbes, and the microbes are respiring some sort of organic carbon. And when they do that, they will reduce their terminal electron acceptors to make iron 2 and or sulfide. So here we have organisms respiring and creating iron 2. Or down here we have a sulfate reducing organism reducing sulfate, producing sulfide. And the iron 2 and the sulfide then goes on to deposit, and either by adsorption and or co-precipitation, to form a new phase. And so that in these figures, this is represented here and here and over here, here and there. And it's there that the contaminant degradation, represented here, say by TCE, it's reduced here to produce products. And here's TCE, it's reduced here to produce products. So the key thing that's the distinction that's being demonstrated here is that the contaminant degradation is remote from the microbiology. The microbiology is producing the iron is producing a new reducing phase, and it's that reducing phase that's degrading the TCE. If the microbes were degrading the TCE directly, that would be biodegradation, it wouldn't be ISCR. The way this figure is set up, the top row represents 2 cases where we have iron 2 here and here sorbed to a mineral phase, in this case goethite, in this case magnetite, to form a new material which is a better reductant of contaminants. In the bottom row here, we're precipitating 2 new phases. Iron 2 and sulfide are producing iron sulfides, and in this case iron 2 is precipitating green rust, which both in those 2 phases are pretty strong reductants. The other thing to note here, we'll come back to in just a second, is that the way this diagram is drawn, TCE is always going to acetylene or something, and it's not going to chlorinated intermediates that are the so-called stall products that are problematic. And that's consistent with the idea that that this is a abiotic pathway. All right, I wanted to reinforce that this idea that iron 2 sorbing onto minerals, so I'm going back 1 slide now, that these top 2 cases here and here, that this is really quite important. And I can't find a real good sort of meta data summary figure for this, but so I used 2 raw primary data figures from the literature. This one on the left is carbon tet, this one on the right is RDX. And what you see here is that carbon tet is not degraded significantly by aqueous iron 2. Nor is it degraded significantly by suspended goethite, which is just iron 3. But if you put the 2 together, carbon tet is quickly degraded, reduced in this case to chloroform. And then over here, if you take RDX and put it in a suspension of magnetite, and you add little or no iron 2, you get little reaction. But as the amount of iron 2 goes up, the reaction rate gets faster. And so clearly this idea of adding iron 2 and having it interact with the mineral surface to supercharge the reactivity of the phase is a strong effect, and it's very important, and it's an area that there's a lot of interest in figuring out now how to better characterize it, because it's a little tricky. Okay, so now on to core concepts. The abiotic reductants. So these are the engineered reductants. So ZVI is clearly the one that's first on the list. I already introduced the idea that DVI is iron 2 would be the other analog. This has been referred to, I think, by Dick Brown as soluble iron. I'm still waiting to find a vendor that's selling some sort of chelated stabilized version of DVI for in situ reduction, but that presumably will be available soon, but off the top of my head, I couldn't think of one. Sulfide, you could add sulfide. People do add polysulfide. There's a lot of applications along those lines. Dithionite, we've talked about, is something that's been used in the field. And then in the context of palladium catalyzed hydrogenation, people people would do is actually add hydrogen directly. There's a few other possible chemical reductants, but they're not well developed at this point. So looking over here now at this version of a redox ladder, this is similar to the previous one except slightly expanded. The one thing I did here at the bottom is I let the scale extend down here to make enough room for zero-valent zinc. You need to notice zero-valent zinc is a lot stronger reductant than iron, which in turn is a lot stronger than all of these intrinsic biogeochemical mineral phases. And also notice that all of these guys here are pretty low on this ladder relative to these organics, reducible organics, and or these reducible metal oxyanions. And so as a general rule, the thermodynamics are fairly favorable. Certainly for the engineered reductants, iron zero, it can reduce any of these things. And certainly zinc can do that too. Notice that the gray zone here, if you can see that on the resolution of your monitor here and here, is the boundary for water stability. So down here, this would be reducing water to hydrogen, and only iron and zinc do that. These other phases don't do that. Okay, so now I'm going to adjust this a little bit here, and I'm going to continue going through some of these core concepts, but I'm going to do them from the perspective of sulphonated ZVI. So we've been working on sulphonated ZVI a lot recently. A lot of people are interested in it. And so I thought it would be attractive for many of you to hear about these core concepts presented specifically using sulfidation as kind of the framework for that. And so again, I'm going to be building off of a recent publication of ours. In this case, it's a review paper that we did it for ES&T last year. And the lead author on this is Damin Fan. Ying Lan contributed greatly, and a number of other authors also contributed significantly to the applied side of that. Again, here's a QR code that will be available again at the end of my presentation. So first of the core concepts, to introduce the kinetics aspect of that, I have this generic version of what we call the standard kinetic model. So for contaminant removal by any mineral phase, but particularly ZVI, we assume that the reaction order is first order with respect to the contaminant and also the metal, and so that's quantified as surface area concentration or mass concentration. That's what those two symbols represent. And those two things are related to one another via the specific surface area. So the density of the reactive surface sites is proportional to the specific surface area. So now you take these things and they're related to one another by this intimidating equation, Km equals KSA times specific surface area. And if you take the log of that, you get parallel contours that are shown on this graph here, where the contour intercept is equal to the specific surface area. So this is log KSA on the x-axis, or y-axis, and log Km on the x-axis. And then again, these contours represent the specific surface area. So this plot is very useful because it's quite flexible. We can put any kind of, most data can be put onto this graph. And so for the sulfidation review, Ying Lan compiled a whole bunch of data. This is essentially all the data that was suitable for this kind of analysis. And we have it here showing for carbon tet and for TCE. And so now the first thing to notice is that the carbon tet numbers as a whole, just to look at the whole cluster of colored dots, lie further up in the upper right corner than the TCE numbers, which lie a little bit lower towards the lower left. The X and Y scales are the same. So what that tells you is it tells you that as a general rule, carbon tet reacts faster than is reduced more easily than TCE. This is actually more significant insight than it might seem at first blush, because note that there's plenty of overlap between this cluster here and that cluster there. So you can cherry pick data and find cases where it would look like TCE reacts faster than carbon tet. But as a general rule, that's not right. As a general rule, carbon tet reacts about 2 times faster by reduction than TCE. Okay, so now you can go further and look a little bit more carefully at the colors. So here the micron size ZVI is represented in blue. Iron oxides are represented in orange here, and NZVI is represented in purple. And blue, or sorry, green here is what we're calling impure ZVI. That would be construction grade scrap iron type materials. And you can see, for example, that the iron oxides and the construction grade materials generally fall lower on this curve, so their reaction rates are slower. And the high purity irons, the nano ZVIs, plot higher, and the reaction rates are higher. And that's true actually in both cases. Anyway, the point of all this for the sulfidation review was the new data that are annotated on the slide that are in these non-circle symbols here. So the purple here, the triangles here, the diamonds here, etc. And what you should take from this is that in all cases, the symbols lie on the upper right direction relative to the corresponding colored symbols that are the transparent circles. These are the sulfidated data, and so it clearly shows that as a general rule, sulfidation makes the dechlorination go faster, although again, there's quite a lot of overlap. So it's not always the case that any particular comparison will show a dramatic improvement in the rate of dechlorination due to sulfidation. But as a general rule, it's probably fair to say that it's favorable for dechlorination. There's an aspect of this that I want to emphasize that that's shown really conveniently with this series of photographs that are shown here, and there's a corresponding YouTube video that goes with this that you might be interested in watching that Serta posted for us. And here what we've done is we have taken a colored dye, which is now basically a surrogate for a contaminant, but such that you can actually see the reaction when the color changes, and also so that it reacts faster. And what we do is we have this colored dye in a syringe that's connected to a port in a column that contains aquifer material that has been conditioned to be fairly strongly reducing condition. And when we withdraw the water from the column, it dilutes the dye like this, and you end up with this lighter blue looking color. Now, the important and potentially surprising result here is that pore water, which by any other metric is quite strongly reducing conditions in that aquifer, model aquifer there, is not reducing this dye. But then what we do is we push the volume of this water into the column, so it goes into the pore space in the column, and then pull it back out again. And when we do that, it comes out pink, which is the color of the reduced form. So the message what this visualizes in I think a very compelling way is that the contaminant needs to contact the mineral phase to be reduced. So it isn't iron 2 in the pore water that's responsible for this, or anything else in the pore water that's responsible for contaminant reduction under these ISCR conditions. It's contact between the contaminant and the reducing mineral surface. Okay, so now changing to the pathway selectivity core concept. I've already briefly alluded to this concept of stall intermediates. You should all be familiar with this. With, for example, TCE, if you do hydrogenolysis on TCE represented by the solid arrows, you can get DCE or vinyl chloride, and they tend to be pretty recalcitrant to further reduction, and so you end up accumulating them in situ conditions where biodegradation dominates. Usually this has been historically has been a big problem. The same thing actually applies to carbon tet. So if you start with carbon tet here and you do hydrogenolysis, you tend to get chloroform, dichloromethane, chloromethane, and these also are effectively stall intermediates, and they tend to be hard to further degrade. In principle, you can push the reaction all the way to completion by following these arrows all the way through, but that often doesn't happen. Instead, in practice, it's more effective to make an end run around those stall intermediates by going around this way. And so this is the reductive elimination pathway for the chlorinated ethenes, and then this is the analogous reductive elimination pathway for carbon tet. In both cases, it gets you to completely dechlorinated products without forming any of these stall intermediates. So what does sulfidation do in this case? Well, it turns out that the data for that is not, there's not a ton of it. And so I had to use the kind of raw data from a couple different journal articles. And so here I picked one from Han and Yan and one from He and Lee. And in this case, this product yield here distinguishes acetylene, ethene, ethane. Over here, they have different palletized irons and then sulphidated iron. And in neither case does this directly quantify the branching ratio to the Stahl intermediates. But in both cases, you note that they're not reporting chlorinated products here, and here they're basically, this is the fully dechlorinated product. That little tiny little bit of yellow there is the only chlorinated byproducts they've seen. So the bottom line is that sulfidation, if anything, helps with respect to the reductive elimination pathway, but the data is not really that clear at this point, and it certainly doesn't hurt it. All right, so the last topic here will be this question of efficiency and demand. We have spent quite a lot of time on this in my lab. This is from papers by Demin Phan when he was in our group. And in this particular case, what we're doing is we're measuring the production of hydrogen, which is utilizing the iron metal, and it's reducing water to make hydrogen. And you can see we're accumulating considerable amounts of hydrogen here. In an experiment where we have untreated nano ZVI. If we treat that sample by sulfidation, we get essentially no hydrogen production. But over here, we've measured the degradation of a contaminant in this system, in this case, another probe contaminant, I2S. And in that case, the rate of the contaminant reduction is about the same. And that's a really important bottom line conclusion about this, is that sulfidation effectively shuts down the hydrogen evolution reaction and doesn't do any real harm or significant harm, and as I said earlier, it might even help a little bit with the contaminant reduction reaction. So that was summarized in this figure here in one of Demin Phan's papers. So here we have nano ZVI electron efficiency. So most of the electrons from the ZVI are going to water, and only a little bit it's actually going to TCE. And when we sulfidated it, it was the reverse. Almost none went to water and almost all of it went to TCE. And I have a brand new figure here from a new paper by Feng He's group that illustrates the same thing. So on the left here, they have sulfidated ZVI, and the right here, they have unsulfidated ZVI. Here they show 92% of their electrons are going to reducing water, and over here, most of the electrons are going to reducing the contaminants. Okay, so very briefly at the very end here, the field scale implications of this efficiency issue has to do with the suitability of using fine particulate reducing materials like NZVI in situ, because there's this question of what's the reductant demand. And we did an experiment some years ago where we did a pilot scale test in the ground, which is represented here. These are little sample wells along this gradient here, and those correspond to these colored symbols here. And this is the direction of groundwater flow, which was from back to front here. And we did an NZVI injection and then pulled water from the sample wells here. This is the nearest one, 0.2 meters, 0.5 meters, and 1 meter. And then you can just visualize as the NZVI breaks through. So here's the water that's unimpacted. Here's the water where NZVI is coming straight through. Further down gradient, that breakthrough happens later. Even further down gradient, that breakthrough happens even later. The really interesting result here is the yellow bottles. So the yellow bottles are NZVI that's been fully oxidized to iron 3. And that's because the aquifer had a reductant demand that basically used up the NZVI and produced basically only iron 3 oxides. And so this is really an important question for sulfidation. We haven't done an experiment like this for sulfidated NZVI, but one would expect that that would help protect the reducing capacity of the NZVI, and the result would be you get more stability of the reductant and in principle a more effective treatment scenario. All right, so with that, I'll be wrapping up here. This is just to remind you of the scope of what we did, at the beginning we talked about definitions and such of in-situ chemical reduction. We looked at the technology perspective on this, and then since then I've followed through about half of these core concepts, and there's a whole bunch of other core concepts that we didn't get to, like the role of impurities, role of passivation, oxidative pathways, delivery, etc., which we'll have to wait for another time. Just to acknowledge the funding for this, it's almost all come from CERTIP, ESTCP. I do also have funding from NSF. We had some funding from NESD, which is a Navy environmental technology deployment program for a little bit of our work. I've had some funding from the DOE Subsurface Biogeochemical Research Program that's relevant to this. I've worked on some projects that utilize some of these technologies, most specifically with Geosyntec and Jacobs, and I get materials from just about everybody that's a vendor in this business. And so I have made a kind of a laundry list of the companies that have supplied bottles that are sitting on our lab bench, and we continue to work with all of you guys and get considerable value from that. Just to finally acknowledge the people that did the key work. Demin Fan, who's again now at Geosyntec, is the person that's responsible for the most of what I talked about today. Ying Lan was probably responsible for the next most of the material that I talked about. Heiji Quinn is a visitor from Tongji University, who's the author on a couple of papers that it should be coming out in the next few months that are significant in the area of sulfidation. And these guys over here were the co-authors on the sulfidation review. And with that, I'll leave this slide up with the QR codes for those of you that want to try to look at that, and then we'll turn it over to John.
Okay, thank you, Paul. I appreciate your perspective. We're going to try to segue here a little bit more into some of the commercial applications and talk about the different ZVI products that are available, contrast and compare them, and try to give you an idea of what actually might work when you're trying to do a true in-situ remediation project. So can you click through, Paul, please? Okay, so what I'm going to show you here, if I can get back to the right slide, is this is an example of the different types of zero-valent iron or zero-valent iron materials that are commercially available. And I'm actually going to try to classify these from small to large. So that the top here are soluble materials, which are divalent iron or ferrous ion species, such as CRS that's sold by Regenesis or some other products that are also available. Get a little bit larger, there's NZVI, which is nanocrystalline zero-valent iron. If you look up here, there's a TEM picture showing the NZVI particles. It's you can see they're aggregated. There's small primary particles, but actually it's kind of a cluster of larger particles. You get a little bit larger, we're talking about the colloidal products, which are what Regenesis sells, which are Aqua ZVI and Micro ZVI. These have discrete 1 to 3 micron particles on average. You get a little bit bigger, there's a super sac over there on the right showing you commodity iron products, which are typically somewhere about 50 to 100 microns in size. They don't look real big. You can actually kind of feel like flour if you put them in between your fingers. And the really big stuff will be cast or scrap iron, such as that car that's being crushed right there. So there's different ways to put these things into the ground. And these include, for the smaller products, you have some options. You can either do put these things in the ground through screened wells, which is a really enabling technology if you want to go into a site that's deep, you want to go through existing wells. You can also put the smaller particles, which are the NZVI colloidal particles, in the ground using DPT. And if you want to use the larger particles, such as the commodity iron, you're more or less relegated to DPT or soil mixing. Paul, could you advance the slide, please? Okay, there you go. There's some pictures. There's on the right, there's a screen well. on the top where the nano iron can be injected into. They can fit right through the slots in the screen. And then on the bottom, there's a DPT type application. So let's expand upon this a little bit further and talk about the different particle sizes that are available. We're going to start here on the left with the soluble iron, which is really not technically a ZBI, but it's similar, so we'll include it in the discussion. And as we go to the right, we're going to progressively larger size particle materials. So let's talk about kinetics. As Paul talked about earlier, there's his KSA philosophy where things that have more surface area generally react more rapidly, and that's generally true. So for the ZBI products, NZBI will probably react the most quickly, and the powdered microscale iron will react more slowly. There's another flip side to that. It's longevity or persistence. Stuff that's more reactive obviously is going to be reacted away more quickly, so it's not going to last as long in the ground. So you might actually have a pretty short-lived remediation program. If you look right here in the middle, there's kind of a sweet spot where the colloidal ZBI is located, somewhere around 1 to 4 microns. That kind of gives you we found over the years of experimentation, kind of the best balance between longevity and reactivity. You actually get a material that's suitable for both. We're going to talk about here is transport. This line's a little bit messier, but something that's higher up is easier to inject, and something that's toward the bottom is more difficult to inject. And obviously, a soluble iron, such as CRS, is easy to inject. It's fully soluble. Basically, it can be injected any place where you can inject water. But if you notice, there's a little dip here at NZBI, and you think, well, these are small particles. They're going to be really easy to inject in the ground, but the science has improved since the early days. What the problem with NZBI particles is that they're so small that they tend to agglomerate. So instead of injecting discrete individual particles of nanoparticles that are 2 microns, you're actually injecting a cluster. My analogy I like to use is instead of injecting a single grape, you're injecting a bunch of grapes. And because of that, you have problems with distribution, and generally your ROIs aren't as good as they are if you actually make the particles a little bit larger. Somewhere around a micron or 2 it appears to be the best size for being able to get a good suspension of the materials, and I'll actually be able to inject them at low pressure through the pore throats of the sand to get a good plug flow distribution. Once you get bigger up, then I would say maybe 6, 7 to 10 microns, you run into that problem where your particles are actually larger than the interconnected pore velocity, porosity in your sand, and you end up having to do fracturing or some sort of fluidization of your sand, or some disruptive process for your soil, and your distribution goes down. And if you have the larger materials such as scrap iron, generally those have to be soil mixed or trenched, which are viable methods in some cases. So I want to talk about the Regenesis product line very briefly. There's 3 ZBI products that are currently available. One of them is Aqua ZBI, which is an aqueous-based suspension of sulphonated iron. So we take iron, we process it, we add proprietary dispersants and organic additives, and we also sulfidate it in situ. So when you buy this product, you get a 40% ZBI suspension in water that's sulfidated, that is very, very, very reactive with chlorinated ethanes and some other groundwater contaminants. This can be co-applied with plume stop, colloidal activated carbon, if you want to have a mixture of absorption and abiotic degradation. Micro ZBI is more of a legacy product. It's not sulfidated, and the way we prevent the reaction with water is to use glycerol as our medium. Glycerol is a food grade, biodegradable donor that's non-reactive with iron, and that allows us to keep a stable suspension, also at 40%, 2 to 3 microns in the glycerol. These both have a viscosity as shipped of about 2 to 3,000 centipoise, and the consistency of paint. Generally, micro ZBI is co-applied with 3DME or other sorts of organic amendments and dechlorinated microbes. CRS is a ferrous iron product. It is an organic compound that has a divalent iron molecule or atom in it. When that what that does is you co-apply that with your organic donor, and you get in-situ reactions that form reduced sulfides, reduced oxides, oxyhydroxides, the species that Paul talked about earlier, and that will also stimulate both abiotic degradation and possibly some enhanced bioremediation. So just briefly, how are these used in situ remediation? There's a lot of different ways to do it. Generally, three different options. One is ZBI alone, which would be straight abiotic, and maybe a little bit of biogeochemical reduction. The other is to co-apply it with organic amendments. And our products can be co-applied. They put in the exact same mix tank, apply to the same pressure, so they get delivered together, and so you have a true synergistic effect. The other option is that colloidal ZVI is at a similar particle size to colloidal activated carbon. They can also be injected together, where you're going to have a great synergistic combination of absorption and abiotic degradation. So with that, I am going to pass this back to Dane, and he is going to take some questions, and we will do our best to answer them.
All right, thank you very much, John. Yes, as John said, that is going to conclude the formal section of our presentation. And at this point, we would like to shift into the question and answer portion of the webcast. Before we do this, just a couple of quick reminders. First, you'll receive a follow-up email with a brief survey. We really appreciate your feedback, so please do take a minute to let us know how we did. Also, you will receive a link to the webinar recording as soon as it is available. All right, so let's go ahead and circle back to the questions. The first question here is for Paul. And the question is, what is a stronger biogeochemical reductant? And they give three options here: ferrous sulfides, ferrous hydroxides, or ferrous oxides.
So you can hear me, correct? Well,
yep, we can hear
you. That's actually a difficult question. Ferrous sulfide, ferrous hydroxide, and ferrous. Read what the other one was.
Basically magnetite.
So I'm hesitating because we're actually working on this right now, and and the more we work on this, the more subtle the problem becomes. I guess a couple of things I can say as generalizations that are certainly going to be true. One of them is that when you compare the numbers, the iron sulfides almost always are the fastest. They reduce dechlorinate, say TCE, faster than anything else. The one thing that's surprising is that magnetite tends to be slow, and iron 2 added to an iron 3 oxide like goethite is surprisingly actually faster than magnetite. Maybe beyond that, I can't give a more simple set of rules.
Okay. Sounds good. Thanks, Paul. Let's see. Next question here, also for you, Paul. The question is, does ZVI reduce sulfate in groundwater?
So that's an interesting question. It's actually a very good question. And when we first started working on ZVI a long time ago, I got I invested a fair amount of time into that problem, because thermodynamically, that is definitely a favorable reaction. And so we thought, wouldn't that be cool if we could do abiotic reduction of sulfate to sulfide? But there is no evidence that that reaction occurs. So thermodynamically, it's favorable, but kinetically, it doesn't happen.
And this is John here. I'm going to put in my 2 cents from a practical perspective. It's actually a good thing that doesn't happen, because there's a lot of sulfate in groundwater, and that'd actually be a pretty high reductant demand, and you'd be using your iron to reduce 500 milligrams per liter of sulfate instead of the 500 ppb of TCE, which you're actually trying to address. So from a practitioner standpoint, it's actually good that it doesn't reduce sulfate.
Okay. All right, great. Thank you, guys. Let's see here. Next question is another question for Paul. And the question is, how much water is reduced to hydrogen by, this is an acronym, but I think what they're saying is sulfadated nano zero-valent iron. So how much water is reduced to hydrogen by sulfadated nano zero-valent iron?
Well, the generalization here is that unsulfadated iron is fairly inefficient in that the majority of the electrons actually go to reducing water and not reducing contaminants. When you sulfadated ZVI, that balance shifts, and so it's actually more favorable to reduce the contaminants than to reduce the water. Putting exact numbers to that is a little bit TBD. The data are just sort of coming out now from different groups about what that amounts to, but I think from what I can recall, the sulfadated ZVI, the amount of water reduction goes down to like 20, 10, 1%. So really pretty low.
Okay.
And from a practical perspective, that's great too, because when you're spending money on a reductant, you want it to go to your contaminant, not to water.
Okay. All right, sounds good. Thank you, guys. Let's see here. This will probably be our final question. This question is for John, and it's regarding Aqua ZVI. It's what is the viscosity of Aqua ZVI when it is injected into the ground?
Okay, that's a great question, Dane. So the way this works is that when the material is shipped, it comes as a fairly viscous suspension, like I said earlier, 2,000 to 3,000 centipoise. But when it's diluted in water at field applications, it's typically somewhere about 1 to 2% of the material in your mix tank. And when it's mixed and suspended, the viscosity is essentially the same as water. So injecting an Aqua ZVI or Micro ZVI product or most colloidal remediation amendments, the real benefit you get out of that is that you're basically injecting water, which means that you can use a simple pump such as pneumatic diaphragm pumps, and you don't have to get into the realms of multi-hundred PSI injections.
Okay, great. Maybe time for one more here. This is also another question for you, John, and the question is, does ZVI reduce the stall COCs like DCEs and VC in groundwater?
I will defer this to Paul. I'll try to give it basically the answer is yes, but slowly. You can do a treatability study of sulfadated ZVI with CIS, and it will go away. I would say the half-lives are probably 2 to 3 times is what they would be with TCE, but there is certainly abiotic degradation. There are some compounds that are basically recalcitrant. Dichloromethane is basically recalcitrant, and 1,2-DCA is also recalcitrant. Those are the 2 compounds that if you have them in your groundwater, it's strongly recommended that you do some sort of biological process using dehalobacters or some other sort of engineered anaerobic bacteria. Do you have a comment on that, Paul, or not?
Well, yeah, just to reinforce that. So the advantage of CVI is not so much that it degrades the stall intermediates like DCE, like John said. It does sort of, but that's very slow. The advantage is it doesn't make them in the first place because it goes by the other pathway, so it avoids making those intermediates. So you have a site where there's in-situ biodegradation that's already produced a bunch of DCE and vinyl chloride. ZVI in principle could be useful, but that's not an ideal application.
Yeah, that's correct. What you'd use ZVI in that case is to actually get the groundwater more reducing, more deoxygenated, and make the environment more favorable for the microbes. But in that case, the primary degradation pathway would be biological.
Okay, all right, got it. Let's see here, maybe time for one last question, and that is, I'm not sure which, I think this might be a question for John, but maybe for both of you. It's, can you address the feasibility of injecting chemical reductants into fractured bedrock? Any success with treating contaminants that have been sorbed in the rock matrix?
And I'll answer that question. There's been colloidal products that have been injected into bedrock. I'd say at least the number of the injections is at least in the tens. the material, the advantage of a colloidal material is that it doesn't sink. They're buoyant, or they sink very slowly. So they're not going to fall down to the bottom of your injection well or your tooling, so you can get them out into a formation. I mean, obviously fractured bedrock has got large channels, so it's not that difficult to get it there, but you got to keep it suspended before it goes in there. Sometimes the challenges with bedrock would be, using packers or some other engineered injection techniques to make sure that you get good vertical distribution and also horizontal distribution. If it's saturated, you're going to be injecting into a saturated zone, which is incompressible and doesn't work too well. But you can give me a call, or the RRS crew could also help you with that if you have a project related to bedrock.
Okay, great. Well, thank you both very much. That is going to be the end of our chat questions. If we did not get to your question, someone will make an effort to follow up with you. To learn more about Dr. Paul Tratnyek's research, please visit the website of the Tratnyek Research Group, or you can access the resources from the QR codes in the recording of this presentation. If you need immediate assistance with a remediation solution from Regenesis, please visit regenesis.com to find your local technical representative, and they'll be happy to speak with you. Thanks again very much to our presenters, Dr. Paul Tratnyek and Dr. John Freim, and thanks to everyone who could join us. Have a great day.